the class 12. So, hope you people are enjoying at your home, being safe, and yes, you people have got your dose of vaccine, and yes, lots of challenges are there in life, but yes, we have to always focus on our goals, and we do continue with our works, whatever the conditions may be. And so, today we are going to discuss one topic that we are actually left or we have not discussed in detail. Chapter biotechnology, principle and processes we have completed, but in that topic, that is cloning vectors. Okay. See, we have discussed about cloning vectors, not like we have not discussed cloning vectors. Okay, what is their role, how they can be used, but there are some characteristic features which you will see in the case of cloning vector that need to be clear. Okay, so what we will do one by one, we are going to discuss the special features of the cloning vectors, what are the characters, what are the properties they need to be have. So the first thing is that we are using the term cloning. Okay, cloning vectors. What do we mean by cloning? See, when we do multiple copy of anything that is known as same structure having same feature is known as cloning. What are the clones? In the case of asexual reproduction, we see the offspring, we can say them as clone because they are almost same. Yes, same thing here also. When we talk about cloning vectors, actually they are which we can say extra chromosomal or non-dependent chromosomal materials. Okay, plasmids are non-chromosomal or non-chromosome dependent materials. They are also extra chromosomal cells, we can say, not cells, means vectors which can be used so that our desired gene can be inserted or incorporated into them and we can transfer them in our desired ones. What happens in cloning vector? Okay, so there are a few examples given in your book, that is PBR 322. It is a type of vectors, but in the introduction, okay, in the introduction of cloning vector, what we are going to see is that example, bacteriophage. Okay, bacteriophage can infect a bacterial cell. Yes, bacteriophage come under virus, right? So, and they can infect bacteria. So you can see that a bacteriophage when enter, okay, a bacteriophage when enter in a bacterial cells, okay, when enter in a bacterial cells, what it do? It transfer its genetic material inside this, okay, and this genetic material get form multiple copies, okay, form the multiple copies inside the host. See, one more thing, the copy number that we used to say is actually the copy of the clone, that plasmid, okay, extra chromosomal material, okay, that is going to be prepared or formed inside a host cell. So cell to cell the copy number vary. Okay, when we are going to choose a host, we also keep in mind that what is the copy number of that particular host cell for that organism. So means for the vectors, Okay, so these things we need to be considered. Now, when you can see that there are numbers of vectors, or we can say plasmids whose copy number can be 100 to 1000, range may vary. Okay, so these are the some features. That copy number is a very much important thing, because what happens, see, when these vectors get enter or plasmid get enter inside the host, so when cell going to be divided, okay, when cell going to be divided, with that what will be happen, these vectors or plasmid also going to be divide inside the cell depending upon the copy number. So as the number of host cell going to be increased, what will be happen, the number of copy cell plasmid is also going to be increased depending upon the copy number. Suppose that copy number is 20, we can say example, copy number is 20 of the plasmid inside a host. So what will will happen when this cell going to be divided from 1 to 2? You can see this 20, so 20 and 20. From the 2 you will get 40 plasmids. So on in this way, copy number vary from cell to cell. Okay. So these are the few features. Okay, what will happen? The bacteriophage is a, we can say bacteriophage act as a vectors. Okay, act as a vectors inside the host. So we can incorporate that inside the bacteriophage, and it is going to release its genomic material. When the bacteria are going to divide, it is also going to be increase its copy number. It is also going to be divide. So this is the concept. Okay. Now, what are the main features that need to be discussed? See, there is few important sites will be present in the case of vectors. What are those? The first of all is your origin of replication. Okay, origin of replication. Origin. Okay, it is known as origin, known as origin of replication. So when you talk a structure of a, suppose this is the structure of a plasmid, so what will be happen? They need to be replicated, then only their copy number they are going to be multiplied. So there must be some position, some place in this from where the process of replication starts. So origin or origin of replication is sequence. It is sequence from where the process of replication start. Okay, and it is also place a reason, we can say, where foreign gene can be incorporated. Okay, so what happened if at this region we incorporate our desired and follow the foreign gene? So during the process of replication, as the number of the sequence going to be increased, what will be happen? The number of that gene also going to be increased. So there must be, I mean to say a plasmid, a vector should have a origin site for the replication process, so that its number can be increased, copy number can be increased by the process of replication, or simply we can say it is the site from where replication starts. Now next is the next is your selectable marker. Now, this is very much important features which should be present in a plasmid or in a vector, because we can say selectable marker is a sequence on the gene of the plasmid, or the sequence which shows the resistance to some particular types of antibiotics. Okay, so selectable markers are the sequence, okay, of the gene which have the which can express the resistance against some particular antibiotic. Why that is necessary? Because the purity of our vectors depend upon this. How can? Because suppose that a particular vector is a resistance to a particular kind of antibiotic. So when we are going to use that vector in that antibiotic, in the presence of that antibiotic does not going to harm the vectors, does not going to kill the plasmid, okay, or does not going to change its genomic sequence and whatever you can say, okay. So that is the role of selectable marker, that we can say they are the sequence, they are sequence which provide resistance to plasmid against a particular antibiotic. Now see, the selectable marker can be more than one or many in a particular vectors. Okay, in the vectors you can get more than one to selectable markers, and I told you that they provide resistance against a particular antibiotic. Okay. Okay. Now the next thing is that see, in the case of selectable marker, suppose just take the example of E. coli. So in case of E. coli, the plasmid present, okay, the vector present, that is BAM H1, okay, the ampicillin resistance, tetracycline resistance, yes, tetracycline resistance, ampicillin resistance. So these are the antibiotics which against which the plasmid show resistance. Okay, so they are the selectable markers. The third thing is that cloning site. Now, the cloning site is the most important. Actually, it is the site, we can say, where generally we are going to incorporate our desired gene. Okay, incorporate our desired gene. First of thing, keep in mind. Second thing, we all know that before incorporating any or integrating any desired gene, we need to cut that gene with the help of restriction enzyme from the desired organism, and to transfer that gene inside the plasmid also, inside the host also, we need to treat that vectors with a restriction enzymes. Okay. Now, thing is that a vector is very much suitable. Note down the point. A vector is very much suitable if it is having single recognition site. Recognition site to treat with restriction enzymes. Why I am saying this? Because the less the number of recognition site, the less the fragment will be there in the host, because plasmid. Because if the restriction in the cloning site, okay, recognition site is more than one, there will be chances of many fragments, yes, and it will be critical for incorporating the desired gene. So it will be very much better if we select or choose a single cloning site in a vectors, okay, so that the fragment would be less number. So what we do for this? See, I told you that in the case of E. coli, PVR 322, there is antibiotic resistance gene, that is selectable marker, one for tetracycline and another one for ampicillin. Okay, one for tetracycline and one for ampicillin. Suppose that from here to here, okay, from here to here, the sequence is responsible for the tetracycline, and from here to here it is responsible for ampicillin. actually what we do in the case of chronic site, see this plasmid is resistance against two antibiotics. What we do by using restriction enzymes, we make cut here. Okay, suppose that we cut tetracycline sequence and we remove this.